All right, let's zoom way in, down to the molecular level to meet the tiny architects that are literally building and running our bodies as we speak. They're the workhorses inside every single one of your cells, and they're called proteins. Let's dive in.
So here's a question for you. What could possibly connect something like your hair, the power in your muscles, and even the way you form memories? It sounds kind of like a riddle, right? But the answer is the same for all of them. Proteins. They're absolutely fundamental to just about everything our cells do. Understanding them, well, that's like understanding one of the core secrets of life itself.
So here's our game plan, our construction plan for this whole explainer. We're going to start with the absolute basics, the building blocks. Then we'll see how they're put together to create these incredible complex structures. After that, we'll figure out how that structure gives them a job to do. And finally, we'll look at what happens when that construction goes wrong, and, what that actually means for our health.
Every great structure, every skyscraper, starts with its raw materials, right? Well, for proteins, those materials are a set of 20 tiny molecules known as amino acids.
Okay, so this is the basic blueprint for every single amino acid. They all have this same core structure, this backbone. But the real magic, the part that makes each one different, is that R group, the side chain. You can think of it like a unique charm on a charm bracelet. There are 20 different charms, and each one gives its amino acid a totally distinct chemical personality. And those 20 different personalities, they can be sorted into 4 main groups. Some are acidic, some are basic, some are polar, and some are non-polar, which just means they really don't like water. Now, this is the crucial part. You got to remember this. This mix of personalities, this chemical diversity, is the entire driving force behind how a protein twists and folds into its final working shape. And here's a cool little detail that just shows you how precise biology is. You know how your hands are mirror images of each other? Well, amino acids come in left-handed and right-handed forms too. And for reasons we're still figuring out, life on Earth has decided to only use the left-handed version to build proteins. It's this incredible level of standardization that makes sure every single protein is built to the exact same specs every single time.
So, we've got our ingredients. How in the world do we get from a simple list of parts to a complex, functioning machine? Well, it happens in 4 distinct levels of architectural design.
We're basically going to build a protein from the ground up. We'll start at level 1, the primary structure, and just keep adding layers of complexity with each step.
First up, we have the primary structure. The best way to think of this is just beads on a string. It's the specific one after the other sequence of amino acids. And this sequence is not random at all. It's spelled out letter for letter by the code in our DNA. A single mistake right here, one bead out of place, can have huge consequences later on.
Okay, next, that long string of beads starts to fold up in certain spots. This is the secondary structure. The 2 most famous shapes here are the alpha helix, which looks just like a coiled spring, and the beta sheet, which looks kind of like a folded, pleated fan. These shapes are held together by relatively weak hydrogen bonds that form along the protein's backbone. And now we get to the tertiary structure. This is where all those unique side chains, those little charms we talked about, finally get to shine. They start interacting with each other, forming this whole toolkit of different bonds, some weak, some really strong. And it's this complex web of pushing and pulling that locks the entire protein into its final, precise, 3-dimensional shape.
But wait, there's more. Some proteins are team players. The final level, the quaternary structure, is when multiple fully folded protein chains, we call them subunits, come together to form a bigger multi-part machine. A perfect example of this is hemoglobin, the protein in our blood that carries oxygen. It's actually made of 4 separate subunits all working together in perfect harmony.
Okay, so we followed the blueprint. We went from a simple chain to a complex 3D assembly. Now it's time for the big reveal. What do these finished structures actually do?
This final, perfectly folded, ready-to-work form has a special name. It's called the native structure. This is the entire goal of the folding process. Out of trillions of possible shapes it could be in, this is the one specific conformation that allows the protein to do its job inside the cell. And this is where it all clicks. We see this incredible link between shape and function. Why is keratin so great for making tough hair? Because it folds into a long, fibrous shape. How does collagen give our skin its strength? It forms this amazing triple helix structure. You have proteins that fold into hollow barrels to make channels, and you have the intricate machine of hemoglobin carrying oxygen. In every single case, the job is defined by the shape. Shape dictates function. But what happens when this incredibly precise process fails? This is where our story takes a bit of a dark turn, because a misfolded protein isn't just useless, it can be incredibly dangerous. So when a protein loses its special 3D native structure, we say it's been denatured. It basically becomes this unfolded, tangled mess, and it can't do its job anymore. This can be caused by things like too much heat or a big change in pH. The protein is essentially broken, and worse, it can start clumping together with other broken proteins.
And when these misfolded proteins start clumping up, they can form these really dangerous things called amyloid fibrils. These are sticky, insoluble clumps that build up in our tissues, gum up the works of our cells, and are at the very heart of many devastating diseases.
Now, thankfully, our cells are not defenseless. They have this really sophisticated quality control system called the unfolded protein response. When it senses that there are too many misfolded proteins hanging around, it sounds the alarm. It starts making more helper proteins to fix the problem, and it slows down the production of new proteins to ease the burden. And if that doesn't work, it tags the bad proteins for destruction. It's a constant battle.
And this constant fight between proper folding and misfolding, this isn't some abstract drama happening in a textbook. It has very real and sometimes very serious consequences for our health.
So many awful diseases are, when you get right down to it, diseases of protein misfolding. I mean, look at this list. Alzheimer's, Parkinson's, even type 2 diabetes. They are all linked to that toxic buildup of amyloid fibrils, just in different parts of the body, like the brain or the pancreas.
And sometimes it all starts with the tiniest little mistake. A condition like osteogenesis imperfecta, brittle bone disease, is often caused by a single wrong amino acid in that initial beads on a string sequence of collagen. Just one tiny error prevents the final collagen structure from forming correctly, and the results for the body are absolutely catastrophic.
It all comes back to this one beautiful, fundamental idea, captured perfectly right here. That simple one-dimensional code, that string of beads dictated by our genes, contains all the information necessary for the protein to fold itself into a complex, functional, three-dimensional machine. It's all right there in the sequence.
So let's end with this thought. Every single second, your body is performing this incredible feat of molecular origami trillions and trillions of times over, with an accuracy that is just mind-boggling. It's a process of immense complexity and just elegance. And it really makes you wonder, in a world where we struggle to build anything perfectly, what could we learn from the blueprint of life itself?